Hello there. Welcome to the first ever lecture for Clinical Chemistry 1, and that is the Introduction to Clinical Chemistry. At the end of this lecture period, the students are expected to define and enumerate the different areas of interest in clinical chemistry, also identify the common analytes tested and the different biologic specimens used in clinical chemistry, and lastly, to differentiate the two chemistry systems. Before we dwell with the different analytes and all the other things that you need to know regarding clinical chemistry, it is a must that you understand what clinical chemistry is all about. Clinical chemistry class, it is a branch of science which deals with the quantitative science that is concerned with measurements of amounts of biologically important substances, or otherwise called the analytes, found in the body fluids. Other textbook definition of clinical chemistry is that it is a biochemical analysis of our body fluids to detect any analytes that you wish to study or you wish to measure in support of the diagnosis as well as the treatment of the certain diseases. Did you also know, class, that the father of clinical chemistry is Mr. Johann Heller, and he is an Austrian chemist? The primary purpose of a clinical chemistry laboratory is to facilitate the correct performance of analytic procedures that yield accurate, so I said they're accurate and precise information, aiding in patient diagnosis and treatment. We all know that the achievement of reliable results requires that the clinical laboratory scientists be able to correctly use basic supplies and equipment and possess an understanding of fundamental concepts critical to any analytic procedure. It is very evident in our time right now that the clinical chemistry continues to be one of the most rapidly advancing areas of laboratory medicine. With the advancement of the clinical laboratories worldwide, it is expected that the medical technologists would deliver or improve the quality of patient care by releasing accurate and precise results for diagnosis and treatment of the diseases. When we talk about reagents, class, in today's highly automated laboratory, there seems to be a little need for reagent preparation by the clinical laboratory scientist. Most instrument manufacturers make the reagents ready to use already, or it's already placed in a kit. Miss, what are reagents? Reagents, class, these are substances that are employed to produce a chemical reaction when coupled with other substances. In other words, reagents are an integral part of any chemical reaction. Depending on what instrumentation is used, reagents can come in several forms, either liquid, in liquid form, it's either dry, it can be cartridge, or a strip. But if it so happen, class, that you're asked to prepare any reagents, then you should be labeling it with the date and time of the preparation, its concentration, and all the necessary things that must be placed there for the material safety data sheet compilation. Testing by manual methods. By the name itself, manual, it means that you're making the reagents and performing all the tests by your hand. This has been done a lot of times in the past, but as we move forward and as automation emerge, we know that testing by manual methods can be time consuming and it's slow, it's labor intensive because you have to be doing and preparing everything by hand. It may be accurate, but we cannot assure its precision because reproducibility can be another reason for time consuming and labor intensive, and at the same time, there is no standardization when we do testing by or through manual methods. Thanks to technology, we can now test all the analytes and all the other processes through automated methods. Automated analyzers class merely incorporate the methods that are previously used when we do manual methods for chemical analysis. Here are the common benefits of automation. One, there's increased number of tests performed by one technologist in a given time period. Another would be labor cost decrease because there's only one medtech that could be assigned to that certain shift, and that medtech could process more than, let's say, more than 10 samples per time period. There can also be minimized variation in results between medical technologists because there can only be slight variations in the technique. Lastly, there's increased variety of techniques or tests being offered that in result to improve patient care. Here are the areas of interest for clinical chemistry. One would be analytical chemistry. This studies and uses instruments and methods to separate, identify, and quantify matter. In our practice class, the separation, identification, or quantification may constitute the entire analysis or be combined with another method. In the separation, it isolates the analytes. Qualitative analysis, this means that you need to identify the analytes, while for quantitative analysis, it determines the numerical amount or concentration of that certain analyte. Next would be biochemistry. It is otherwise known as biologic or biological chemistry, and it's the study of chemical processes within and relating to living organisms. A subdiscipline of biochemistry is both chemistry and also biology. Biochemistry may be divided into three different fields, that is, structural biology, enzymology, and metabolism. For enzymology and metabolism, we will be discussing it more on clinical chemistry 2. But for metabolism, by the way, it can be incorporated already for clinical chemistry 1. Another would be endocrinology. This is a branch of biology and medicine dealing with the endocrine system, its diseases, and its specific secretions, which is commonly known or popularly known as the hormones. It is also concerned with the integration of developmental events such as proliferation, growth, and differentiation, and the psychological or behavioral activities of metabolism, growth and development, tissue function, sleep, digestion, respiration, excretion, also our mood, the stress, lactation, movement, reproduction, and sensory perception caused by our hormones. Another area of interest for clinical chemistry would be instrumentation or automation. This is a collective term for measuring instruments, and it's used for indicating, measuring, and recording physical quantities. It is also a field of study about the art and science about making measurement instruments involving the related areas of automation, of course. The term has its origins in the art of science of scientific instrument making. Instrumentation class can refer to devices as simple as direct reading thermometers that we have at home, or as complex as multi-sensor components for clinical chemistry analyte measuring devices. Today, instruments can be found in the laboratories, even in the factories, and even for your vehicles, as well as in everyday household use. And lastly, toxicology. This is a scientific discipline which overlaps with biology, chemistry, pharmacology, and even medicine that involves the study of the adverse effects of chemical substances on living organisms. It's also the practice of diagnosing and treating exposure to toxins and toxicants. The relationship between dose and its effect on the exposed organism is of high significance in toxicology. For toxicology class, did you know that it's currently contributing to the field of cancer research? Because some toxins can be used as drugs for killing tumor cells. So that's a very nice must know, no? In this slide here, class, it is the common analytes that we measure in the clinical chemistry laboratory. Those are the ions, so different ions, salts, and minerals. Miss, what are ions, salts, and minerals? Ions, class, can be either carbonate, acetate, ammonium, nitrate, nitrite. Those are ions. But for salts, remember your sodium chloride, potassium dichromate. So we also test for those. How about the minerals? Sure, Jude, you don't know? The important minerals of our body includes calcium, iodine, magnesium, phosphorus, sodium, zinc. Those are examples of minerals of our body. How about the small organic molecules? Example of which are what? It can be glucose, that's for sugar or carbohydrates. Cholesterol, an example for lipids. Stearic acid, and that's another example for a fatty acid. Lysine is an example for amino acid. So those are small organic molecules. While for large macromolecules class, of course, no, there are three main types of biologic macromolecules for the mammalian system. That's carbohydrates, nucleic acids, proteins, and lipids. No, not three main types. That's four types. Carbohydrates, nucleic acid, proteins, and lipids. Make sure you write those examples down in your notes because that will, of course, go out in my examinations. The next two slides are the common analytes in the clinical chemistry laboratory with the different examples for the analytes that we measure in the lab. Aside from the analytes, so we're already acquainted with the different analytes that we measure in the laboratory. We also have the different biologic specimens to use in order for us to test for those different analytes. Let's discuss these different biologic specimens one by one. The most common biologic fluid collected for clinical laboratory testing is blood. For blood class, it consists of two main parts. The fluid portion, which we call the plasma, if it's an anticoagulated blood, while the other portion is called the serum for coagulated blood. And then aside from the plasma or serum, there is the cellular portion where you can see the red blood cells, the white blood cells, and even the platelets. This diagram here shows the difference between plasma and also serum. So again, for plasma, the centrifugation is done directly with samples that have anticoagulants. For plasma class, clotting factors are present, an example of which is the fibrinogen. So that area there on top is the plasma. It is the main component of blood and consists mostly of water with proteins, ions, nutrients, and wastes mixed in. In between the plasma and the packed red blood cell, there is what we call the buffy coat area where you can see the WBC and the platelets. On the other side, class, centrifugation is done after coagulation without anticoagulant. So this means this sample is coagulated. Serum is usually used for diagnosis, so majority of immunology and serology tests, even the chemistry tests, makes use of serum. Another biologic specimen would be the urine. It's suitable for tests that evaluates and checks for kidney functions. This test also look at waste products that are excreted by the kidneys and for metabolites that are cleared quickly from the bloodstream and can accumulate in the urine. An example of analytes that can be tested with the use of urine as a biologic sample could be drugs of abuse. There are also other tests, can be creatinine clearance measurements, makes use of 24-hour urine. So you collect urine samples from the patient for the entire 24-hour duration. So you will learn more about that in clinical microscopy. We can also test urine for proteins and or glucose so that we know if there is already certain amount of damage in the kidneys. So those are the different test examples that could help us diagnose if there are problems with the kidney functions. This diagram here shows the different types of urine sample and the purpose for testing them. Another important fluids that we receive in the laboratory are the amniotic fluid, the peritoneal fluid, the pericardial fluid, and the pleural fluid. An amniotic fluid class is a clear to slightly yellow liquid that cushions the fetus within the amniotic sac. The developing fetus floats in the amniotic fluid for the entire duration of a pregnancy. What could be an indication for amniocentesis? It's either if the mother's age is 35 or more. There can be an indication for chromosomal defects. That's also another reason why the mother will undergo amniocentesis. Family history of chromosome abnormalities such as trisomy 21 for Down syndrome. The parents carry an abnormal chromosome rearrangement, or if the parent is a carrier of a metabolic disorder. And also to assess if the fetus lungs have already matured. So that's for fetal lung maturity. The next fluid would be peritoneal fluid. So the collection is called abdominal paracentesis. And this peritoneal fluid class is collected in the peritoneum of the abdominal cavity, which lubricates the surface of tissue that lines the abdominal wall and the pelvic cavity. This peritoneal fluid is a serous fluid that covers most of the organs in the abdomen. An increased volume of peritoneal fluid is what we call ascites. Another biologic fluid is the pericardial fluid. This is the serous fluid secreted by the serous layer of the pericardium into the pericardial cavity. The purpose for this pericardial fluid reduces friction within the pericardium by lubricating the epicardial surface, allowing the membranes to glide over each other with each beating of the heart. Pleural fluid is a serous fluid produced by the serous membrane covering the normal pleura. So that's the pleura class is the lung lining, the lining of the lungs. So most fluid is produced by the exudation and parietal circulation via bulk flow and reabsorbed by the lymphatic system. When the accumulation of pleural fluid is noted, we can do cytopathologic evaluation of the fluid, such as as well as clinical microscopy, can also be microbiology, chemical studies, tumor markers, and all of the other tests that are required that are for diagnostic, that serves as diagnostic tools for determining the causes of this abnormal accumulation. Because class, they are normally there, the fluid is normally there, but there shouldn't be any accumulation. So if there are abnormal accumulation, class, that's the time that there's an indication for removal of the fluid for testing. And lastly, for our objective or the learning outcome, we need to discuss the two different chemistry systems. Actually, we have the dry reagent strip. This requires the comparison of a color change on the reagent strip color chart. So you will be reading the results with the help or with the aid of a strip or reagent strip color chart. Well, for the wet and dry chemistry systems, this makes use or utilizes a spectrophotometer to mechanically measure any color change. The dry reagent strip method is commonly done in the chemical examination for urine in the clinical microscopy section. While for the wet and dry chemistry systems, most of our automation has already employed these types of system. The main difference between the two is that the dry reagent strip is subject to inaccurate, not really inaccurate, but there is subjectivity to the reading of the results compared to that of the wet and dry chemistry systems because it has been standardized already. What are the types of measurement in the clinical chemistry laboratory? I think I have already discussed the difference between qualitative measurement or analysis. This gives results in descriptive, non-numeric form. So when you make use of the dry reagent strip, class, most usually we make use of this color charts so that we can compare and contrast and give our readings. While for the quantitative measurements, this gives results in definitive numeric form. So it has numbers with the units provided. Thank you so much for listening. That is all for the introduction to clinical chemistry. I hope you've learned something new from me today. If you have any questions, clarifications, you may message me in my Cello account. So God bless us all.